You're going to be
I'll be introducing Eran.
introducing. You want to introduce both at the beginning or just introduce Eran at the second part?
Yeah, I'll do that at the second part.
Okay. All right. So let me start by introducing Robyn Wiseman, who's going to be presenting today. She's got a really cool story to share about the correlation between a dipeptide that's in our brain called NAAG and cognitive function in patients with living with HIV. As background, Robyn completed her undergraduate degree with a double major in chemistry and forensic sciences at West Virginia University. And then she did her master's degree in King's College in London in forensic science. And then she did her master's thesis work at the National University of Singapore. And then she worked for a year as a forensic toxicologist in Seattle. Robyn likes to move around. And then she came to Johns Hopkins to start her graduate work in the Department of Pharmacology and Molecular Science, where she is now. She's a fifth-year graduate student working with myself and Christy Bigos on this translational project. One of the things unique about Robyn is she's doing both a basic science element of this project she's not going to be talking about today, and that is trying to develop drugs that enhance NAG. And then what she will talk today is her clinical work looking at brain NAG levels and its association with cognitive function. So glad to have you, Robyn. I'll turn it over to you.
Okay, yes. So like Barb said, this is a really exciting project. It's very new, so I'll be sharing a pilot study that we completed recently. So just an outline, I'll give you a background on NAG and also GCB2 and why we think it's a good target, why we decided to look at this in HIV, and then some of the results from this pilot study. So N-acetyl-Aspartyl-Glutamate, or NAG, is a neuropeptide that I'm guessing a lot of you probably have not spent much time thinking about, but hopefully today I'll convince you that it's important. So it's highly prevalent, especially in the brain. There's about 100 times more in the brain than in other organs, and it's in a higher concentration in white matter than gray matter. It's produced enzymatically by a series of transferase steps shown on the right side of this slide here. It's produced in neurons as well as glial cells. And the levels of NAG are controlled in vivo by the enzyme glutamate carboxypeptidase 2, or GCP2. You may have also seen it called nalidase, or prostate-specific membrane antigen, because it's one of those proteins that's been discovered independently in different regions of the body. It's a transmembrane zinc-dependent metallopeptidase. It's predominantly normally found on astrocytes, but we also know that it's highly upregulated on activated microglia. So you can see how this starts to become relevant, because in neuroinflammatory conditions like HIV, you have activation of these microglial cells. So the way that GCP2 controls NAG levels is it cleaves NAG into N-acetyl-aspartate and free glutamate. So you've got this additional problem where if you have upregulated GCP2, you're also further increasing your pool of free glutamate, and in an already cytotoxic condition, that's problematic. But the problem here is twofold, because NAG also helps to modulate glutamatergic signaling itself. So it acts as a receptor antagonist at NMDA receptors and also as an agonist at these metabotropic mGluR3 receptors. So we think that NAG is playing a role in helping to fine-tune and regulate what glutamate is doing in the brain. We think its activity at mGluR3 receptors is especially important because in the dorsolateral, like especially the layer 3 of the dorsolateral prefrontal cortex of primates and humans, we have this region that's highly evolved, and there's this recurrent excitation of the neurons. So in the absence of a stimuli, your brain can still hold on to and manipulate information, which provides the underpinning for some of these higher order cognitive processes like working memory and executive function. But you can have this feed forward signaling in this cyclic AMP PKA cycle where you open these potassium leak channels, and that reduces the connectivity between these neurons. But mGluR3 can downregulate or inhibit this adenylyl cyclase activity, so you have less cyclic AMP, you have less of this signaling, and the neurons are communicating more efficiently. So the way that NAG is playing into this, if you have GCB2 inhibition, you're increasing your pool of NAG, so you're having more activity of these mGluR3 receptors, and we would predict greater communication between the neurons. And this has actually been demonstrated in a non-human primate model in the Arnston lab at Yale. So they gave a GCB2 inhibitor, and in this panel, in a dose-dependent manner, you can actually see that they saw increased firing in this region when they inhibited GCB2. So this is a good proof of concept that this seems to actually be occurring. There's also a lot of other preclinical evidence that GCP2 inhibition is a good strategy to increase NAG and also improve cognitive performance. So I mentioned the Arnston lab already. They've done a lot of work in aging. So they've looked in aged rodents like rats and also non-human primate models. And when they treat with the GCP2 inhibitor, they see improvements in cognitive performance, and they especially focus on these higher order domains like working memory tasks. They also have a paper that you should look out for that's coming out soon. They're showing that if you chronically inhibit GCP2 in these aged rhesus macaques, you see reductions in phosphorylated tau levels. There are other models that people have looked at. So some groups have looked at these NMDA antagonist models of schizophrenia. So there's this glutamate hypothesis of schizophrenia that you've got aberrant glutamatergic signaling, which is part of what causes schizophrenia to happen. So if you treat rodents with NMDA antagonists like MK801 or PCP, you get a schizophrenia-like psychosis accompanied by cognitive domain deficits in areas like pre-pulse inhibition, so sensory motor gating, and also things like working memory. And if you treat with a GCP2 inhibitor, you get recovery of that cognition. And recently, a group took a different strategy. So in these neural stem cells, they overexpressed the gene that encodes for the enzyme that synthesizes NAG. So a different strategy, but still with the goal of increasing NAG. And they also saw cognitive recovery in a mouse model of traumatic brain injury, as well as preservation of synapses in the hippocampus. So going into the more translational relevance of this project. So this was a paper that was published a while ago by someone in our lab, Kristen Braun, or now Hollinger. So she did a real mouse to man study in multiple sclerosis. So the right is some of the mouse work she did. She did an EAE inflammatory model of multiple sclerosis. And when she treated with the GCP2 inhibitor that we have, so this is just a potent tool inhibitor that we use, she saw increases in NAG levels in the cortex, and she also saw improved performance in a Morris water maze. And then this is some of the human data. She measured NAG levels in some humans with multiple sclerosis, and she saw positive correlations with some of the cognitive battery they did. I'll go into a little more detail on some of the memory relevant tasks that she did later. Another key paper that's been published in this area is this one that was looking at a missense mutation in the FOLH1 gene. So this is the gene that encodes GCP2. So if you have this missense mutation, it was found that you have decreased NAG levels, and you also it's also associated with lower IQ score. And then this was one of the only other papers that's really been published in this area. So this was looking at NAG levels in schizophrenia. So they were looking at the relationship between NAG, again, measured by MRI, with schizophrenia symptoms, and then also memory scores. And so they found a negative association with the negative symptoms of schizophrenia and a positive association, again, with memory. So I've shown you a lot of data in other disorders. So why did we decide to look at this specifically in HIV? So we know that cognitive impairment is still a big problem in HIV, despite the fact that we have these efficacious antiretroviral therapies that allow people to live longer, healthier lives in a virally suppressed state. So the schematic on the right is showing a review that was demonstrating that despite the fact that the severity of cognitive disorders has decreased, the number of people that have some form of impairment has not really been affected. And the domains that tend to be affected are, again, these higher order domains that are more subtle, like working memory and executive function. So things that are really important for people to be happy and successful, and are also linked to these circuits that are modulated by NAG. Additionally, one of the reasons that we hypothesize that there's ongoing neurocognitive deficits in people that have HIV is that there's glutamatergic dysregulation and excitotoxicity. So these infected monocytes get into the brain, and even after viral suppression, there's this ongoing reservoir of HIV in the brain. So this leads to activation of immune cells. You have increased glutamatergic signaling, reactive oxygen species, cytokines. And like I mentioned earlier, when you have activated microglial cells, you have upregulated GCP2. So you have more GCP2, you've got more of this excitotoxic glutamate, and you also have less NAG to help regulate this. So an outline for the pilots, or some of the methods that we used for this pilot study, we looked at 40 people with HIV. They were all older adults, so they were at least 50 years of age. They had no past 3-month history of drug abuse. They were not testing positive for mood disorders, and they didn't have any history of CNS infection other than HIV. So these people underwent a 7 Tesla magnetic resonance spectroscopy scan. So I'm emphasizing 7 Tesla. This is a really high field strength magnet. It allows the greatest spectral resolution, which is important, specifically when we're studying NAG. I mean, always important to have good spectral resolution, but especially when we're studying NAG, because you can peak here, the largest one in MRS is the N-acetyl-Aspartate peak, and the small shoulder on the side is NAG. So with higher spectral resolution, we're able to get a more accurate separation of the N-acetyl-Aspartate peak from the NAG peak, so we can quantitate and study them separately. These people also underwent a comprehensive cognitive battery, which is great because it lets us get at some of these domains like working memory and executive function that we think are impacted by NAG. So to get into some of the results from the study, so this is in the frontal white matter. Like I mentioned before, the highest levels of NAG are found in white matter. So when we're studying NAG specifically, we always try to measure it in one of these white matter tracks, like the centrum semioval, because it allows the most accurate estimations. So the tests that went into our attention working memory composite score were from the California computerized assessment package. So these tests look at reaction time, working memory, and sustained attention, and they're pretty easy to deliver. And this is a pretty standard method that's used in HIV cognition. But basically, it would be simple things like press a button every time you see a triangle, or only push a button if you don't see a triangle, or if you see 2 triangles in an array. And so we saw this positive association between attention working memory and this frontal white matter NAG. So this is exciting because we're seeing domain specificity. We would expect working memory to be frontally controlled, and we're seeing NAG in that region relating to this score. And this also aligns with data from that full H1 genetics paper that I mentioned to you earlier, where they were looking at recent onset psychosis and healthy volunteers. So they saw in this white matter track, you see a positive association with visual factor memory score. And when they had people do a functional scan where they were doing a working memory test, they saw less efficient cortical activation. So the brain was working harder to reach the same level of performance. So then we also looked at the hippocampus, and we found a positive association between executive function and hippocampal NAG. So our executive function score was made up of this trail making part B, which is where you would go from like 1 to A, 2 to B. So it's processing time, but also you have to remember where you're going at any given time. And then also Stroop interference, which is where you're reading. You're supposed to be saying the color that the word is, which is more difficult when the word itself is spelling out a different color. So this also aligns with data from that multiple sclerosis paper that I mentioned earlier. So they used a completely different measure of executive function. They used the DCAF's card sorting task, but they also saw positive associations between NAG and executive function score and the different measures of this task. So it's exciting that we're seeing not only that like hippocampus seems to align with executive function, but also that like across these disorders, we're seeing the same kind of relationships with NAG, suggesting that NAG has this overarching role in cognition, and it's probably important in a lot of disorders and maybe just healthy cognition. So one of the more novel findings of our study is that NAG in the basal ganglia positively correlated with verbal fluency. So our verbal fluency test was the controlled oral word association test. You're given a letter and have to name as many things as you can think of that start with that letter in a category. It makes sense that we would see this association because the basal ganglia has been studied by a lot of groups in HIV, and they found by MRI changes in the structure and function of the basal ganglia and relationships between that and verbal fluency. Additionally, some fundamental work that's been done in receptor distribution has shown that there are mGluR3 receptors in the basal ganglia. So the target receptor for NAG is present. One of the other interesting things that came out of this study is that there was a negative relationship between NAG and the mesial precuneus. with attention working memory. And so at first, this seems counterintuitive because I've spent this whole 20 minutes talking to you about how NAAG is positively associated with cognition. But if you dig into this a little more, I think it makes sense. So I've showed this very specific relationship between NAAG and different regions of the brain with specific cognitive domains that seem to be when that region is activated. So you've got like working memory and frontal cortex, you've got executive function and hippocampus. So a region like the mesial precuneus that may or may not necessarily be the most relevant for attention working memory, it makes sense that NAAG, sorry, my allergies are terrible, may be playing a different role in this region. And in that full H1 paper that I showed, they also saw inefficient activation in the mesial precuneus in patients with recent onset psychosis. So it's possible that you're seeing more activation of this region when the brain isn't working as well on working memory. So some future directions for this study. So preclinically, like Barb mentioned at the beginning, the therapeutic core is working on developing novel brain penetrable inhibitors of GCB2. So we have several different strategies going on right now. This is a really challenging target because it's an enzyme in the brain that recognizes small polar charged molecules, which don't readily cross the blood-brain barrier. So we have a couple high throughput screen campaigns going on right now, one in collaboration with NCATS at NIH. We've done a DNA encoded library screen, which allowed us to screen several billion compounds at once. And we're going to be decoding that data soon. We've also done a rapid and library screen in collaboration with June Liu here at Johns Hopkins. And there's a few different prodrug strategies like dendrimer conjugation, which will help target the inhibitors to activated immune cells where there's inflammation present, and then different prodrugs to try to take advantage of like transporters to get things into the brain. And so the idea would be that the lead compound that comes out of this would be tested in the eco-HIV mouse model that we have set up in the lab that does show cognitive deficits. And then clinically, because this is just a pilot study, there's a lot more work that we need to do. So we got a concept sheet approved to access the NWCCS data, which if anyone's not familiar, this is a database made up of 2 data sets from 2 large scale, like multi-thousand person longitudinal studies. So this is a really rich data set with longitudinal cognitive data and a lot of other physiological measures. And then since we've only measured NAAG so far by spectroscopy in the brain, we also want to look at plasma and CSF to see what those relationships look like and to see if there are differential biomarkers of cognition in HIV. We also have a lot of ongoing studies in other disorders where there's some level of glutamatergic dysregulation. So we're looking at ADHD as well as mild cognitive impairment in the absence of HIV. So some acknowledgements. Thank you to Barb and Leah, who have really been the leads on this project. I wouldn't have been involved in anything with HIV if they hadn't brought this to me. And then the people that really did the work. And then my department and my lab, which are the reason that I'm here.
All right, very nice job, Robyn. Let's go ahead and open it up for questions. Any questions for Rob? We got a couple minutes before we can turn it over to the next speaker. Let me ask a question, Robyn, that I hadn't thought about until you presented today. Is there other known brain metabolites that correlate as well as NAAG does to cognition? It's striking that you're seeing this correlation between cognition and the levels of NAAG in the brain across different diseases. Is there anything else that shows this? People looked at other brain metabolites with such correlation?
I mean, a lot of people have looked at some of the more mainstream ones like glutamate and GABA, and those and a lot of disorders tend to have like in my ADHD data, we see really strong relationships with glutamate.
Hi, this is Pim Rausch from NIMH. I had a question. So you're starting to make a specific, a case for a cognitive specificity. So to say, you're really indicating that you think this is affecting executive functioning. What I didn't see was that you were presenting data on some other cognitive functions where there was basically absolutely no correlation, so that it is really that specific. Do you have that data?
Yeah, so as part of the cognitive battery that these people underwent, they also had a lot of like fine and gross motor skills tests and some really basic processing speed things. And in those domains, we don't really see a relationship. It seems to be very specific to these higher order cognitive functions like memory.
Thanks.
Hi, this is
Any-
Christy. I was just going to add to that that's been the same sort of story that's played out across other cognitive tasks in other disorders as well. So it's really interesting that these are more like frontally controlled tasks that are more regulated by NAAG.
And then this is Leah, just to chime in, Pim, with the plasma work and CSF, Robyn and, we'll be able to look at more of the RDoC metrics as well. So not just the standard. So we started with what was there, but they'll really get to probe cognitive control and other aspects, social processing and the like.
Great, and please also include mental health.
It's all, we have a ton of that as well. Yes.
Yeah. All right, if there are no more questions, Lee, I'll turn it over to you to introduce the next speaker.
Excellent.
Nice job, Robyn. Really
Yeah.
nice job.
Yeah. Always, you're an amazing speaker. I get the privilege to introduce our second speaker, Dr. Eran Shore, who is an MD and computational neuroscientist who's been serving as a postdoc within our Johns Hopkins Brain Health Program here in neurology. And he's been primarily being mentored by both myself and Dr. Raha Daske, who is not here today. She's on maternity leave. And Eran has been leveraging cutting-edge data science and statistical techniques to delve into the lasting neurological impacts of HIV. And his work looks to unify really computational neuroscience and neuroinfectious disease to better understand and treat neurological disorders. So I'm going to turn it over to him, and he is going to be sharing some of his work. This is the TK and sleep, if you want to share your title. And this has been work that he has been working on within the MaxWise combined cohort study. So I will turn it over to you, Eran.
Thanks, Leah. Yeah, thanks for that kind introduction. And yeah, also it was a great talk, Robyn. Really enjoyed listening to that. So as Leah mentioned, I'm Dr. Eran Shore, and I'll talk a little bit about the tryptophan-Kynurenine metabolic pathway and daytime dysfunction in women with HIV. So when I talk about daytime dysfunction, I'm really speaking about the subjective experience of poor sleep. And we know that sleep is an essential part of both physical health and mental health. And in the context of HIV, it's particularly important when we consider the role of sleep in terms of medication adherence and general quality of life. In HIV, sleep disturbances are particularly prevalent. Next slide. So 58% of all people with HIV experience some form of sleep disturbance. And in the Women's Interagency HIV study, we've shown that women are particularly affected. And women with HIV were 17% more likely to report insomnia compared to those without HIV. So sleep is clearly a problem, and it's a problem that impacts quality of life, impacts physical and mental health, and it's something that we need to really get a better grasp on in terms of what's affecting sleep in the context of HIV. So there's a couple of ideas about what might be the cause. In the past, it was previously thought to be partly a result of efavirenz use. Efavirenz is one of the older antiretroviral medications. It's known to cause a variety of neuropsychiatric conditions and problems, but these days we have kind of shifted away from efavirenz, and we still see sleep disturbances. So this hints that there's something deeper. It's not necessarily the antiretrovirals. Another idea one might have is that in HIV, there's likely to be a lot of comorbid mental illness, and this might be causing the sleep issues. And it's hard to say if this is the case, as disturbances like PTSD or depression are in part defined by the sleep disturbances. So we can't really get to the bottom of whether it's the chicken or the egg. What's causing what? Is it the mental illness, or is it the sleep causing one or the other? So because this is quite an entangled relationship, we try to look at a more biological approach. And we went specifically to the Tryptophan-Kynurenine metabolic pathway. So why this pathway in particular? Well, because tryptophan is a precursor to both serotonin and melatonin. And as you might know, melatonin is a key mediator of circadian regulation. On top of this, we know that people with HIV exhibit disturbances in this pathway and the metabolites along it. What's thought to cause the disturbances in the context of HIV is a change in the activity of the IDO enzyme. So IDO stands for indoleamine 2,3 dioxygenase, and it catalyzes the conversion from tryptophan to kynurenine. So tryptophan can go mainly in one of two directions. On one hand, it can go towards serotonin and melatonin, and on the other hand, it can go to kynurenine and its metabolites. And we know that both HIV-related proteins like TAT and inflammatory mediators like interferon gamma and TNF alpha induce IDO as well. So it's this kind of constellation of an enzyme which is highly active and a deficit in tryptophan, which we think might be underpinning the sleep disturbances. Here is a streamlined diagram of the paradigm we're really looking at. On the left-hand side, we have tryptophan, and tryptophan is an essential amino acid, meaning it's not synthesized endogenously. Our tryptophan levels are entirely dependent on our dietary intake. And furthermore, the rate of absorption and the degree of absorption of tryptophan is dependent on the microbiome, as well as competing amino acids. Once tryptophan is in our circulation, we're looking at these two pathways. So firstly, towards serotonin and melatonin, which is the key determinant of sleep. And then secondly, towards kynurenine and its metabolites. When IDO is induced and tryptophan is shunted towards kynurenine, you have the production of reactive oxygen species, which is potentially one way we find neuronal damage. Another way is through the kynurenine metabolites themselves. So metabolites such as quinolinic acid are directly neurotoxic. So there's a couple of things going on here. It's the production of reactive oxygen species and these kynurenine metabolites on one hand, and on the other hand, it's the deficit of melatonin. So this constellation of concerns really brought us to the aim and hypothesis of this project. Our aim was to compare the relationship between the tryptophan-Kynurenine pathway and daytime dysfunction in women with and without HIV, with the hypothesis being that women with HIV will have a metabolite profile reflective of greater IDO activation, and subsequently IDO activation being associated with daytime dysfunction. So how did we go about achieving this aim? Well, we used participants from the Women's Interagency HIV study. These were women living with and without HIV, aged between 35 to 70 from the New York and Chicago sites of the study. Importantly, we used a very select population of these women. Our population were on stable antiretroviral therapy, excluding efavirenz, which we know could be a confounding factor. We looked at those that had a viral load of less than 200 copies per mil in the 6 months before enrollment. And we further excluded women with diagnosed psychiatric conditions or who were on psychotropic medication. We also excluded those who had diagnosed substance use disorders. So then how do we go about quantifying daytime dysfunction? Well, we use the most valid methods that we have, the Pittsburgh Sleep Quality Index, or PSQI. And this is kind of the gold standard in the field of subjective sleep quality. And one of the subcomponents of the PSQI is daytime dysfunction. This subcomponent is made up of the sum of 2 questions. The first question is, during the past month, how often have you had trouble staying awake while driving, eating meals, or engaging in social activity? And this is scored on a 4-point Likert scale, with 0 indicating the least severity and 3 indicating the most severity. Then the second question is, during the past month, how much of a problem has it been to keep up enough enthusiasm to get things done? And it's scored on a similar Likert scale, and the sum of these 2 give the PSQI daytime dysfunction score. Then moving on to the metabolomics, we used a targeted panel. We performed high-performance liquid chromatography and mass spectrometry. And we thank the Broad Institute for doing the metabolomics work for us. Now to discuss some of the results of the study. Firstly, our sample, we had 141 women with HIV and 140 women without HIV. Our samples were relatively of the same age, with the women with HIV being slightly older, the mean of 55. In terms of ethnicity, there were no differences, no difference in their menopausal status. We did find a difference in their subjective depressive symptomatology. So the CESD is a score that rates subjective depressive symptoms. It's important to note that we had excluded those that were diagnosed by a psychiatrist to have depression, and there were differences between these samples. However, in the CESD, there are questions that are directly related to sleep. So this certainly confounds our interpretation of the CESD. So what we did was we excluded those questions, and we really looked at the emotional as opposed to the somatic part of depression. So by excluding these sleep questions in the total score, we created a CESD affect score, and then there were no differences between the groups. Another thing to note is that in the women with HIV, we found there to be slightly a higher percentage of smokers. In our HIV group, the median CD4 count was just over 700, with a nadir CD4 count of less than 200. So it's another thing to consider that many of these women had dropped their CD4 counts to what previously was HIV stage 4. And then we have this small population, an N of 25, who we defined as viremic. And this is really a relative viremia. If you remember, we excluded all participants that had a viral load that was larger than 200 copies per mole. We defined viremia as those who had viral loads of between 20 to 200. So it's a really low-level viremia which we're talking about. So then to look at some of these results, here we have a heat map showing the Spearman correlation coefficients between daytime dysfunction in various groups and metabolites in the tryptophan-Kynurenine pathway. In this analysis, we controlled for age, menopausal status, the non-somatic depressive scores, and smoking status. So on the x-axis, we have the metabolites, and on the y-axis, we have daytime dysfunction in the different groups: women with HIV, women without HIV, the small population of viremic women, and aviremic women with HIV. The first finding we get from this is that low 5-hydroxytryptophan and low serotonin were correlated with greater daytime dysfunction. And we don't see this effect in the viremic group, although the trend is in that direction. This is likely due to a very small sample size. And this makes sense. Low serotonin likely leads to low melatonin and subsequently increased daytime dysfunction. More interestingly, though, we find these relationships between kynureninic acid and the kynureninic acid tryptophan ratio. Both of these are markers of IDO activation. And both of these are only significant in the population of women with HIV. So here we can really show that greater daytime dysfunction is associated with increased IDO activation in our sample. Another way to consider these results is by looking at the metabolic pathways themselves. And here we're looking within each group and comparing those with versus without daytime dysfunction in the group. We define those with daytime dysfunction to be any participant that scores more than 1 in the daytime dysfunction overall score. Here, the color of the square represents the log-fold change difference between those with and without daytime dysfunction, and the number of bold borders indicates the significance level. So a single bold border would represent a 1-star significance, 2 bold borders, 2-star significance, a 3-star, so on. And here you can really see in those with daytime dysfunction, there's a significantly lower level of these melatonin precursors. And in the group with HIV, we find this higher level of kynureninic acid. And to remember, IDO acts on this pathway. So when we take the kynureninic acid to tryptophan ratio, we really get a marker of IDO activation. So higher IDO activation relates to daytime dysfunction in this population. In the women without HIV, we get results that we expect in that there's lower levels of serotonin precursors in those with daytime dysfunction, but crucially, we don't have the same relationship with kynureninic acid. So there are various strengths and limitations with our study. Firstly, we used a self-report measure of daytime dysfunction, which is certainly prone to reporting bias. However, in this work, we intentionally wanted to get a subjective experience, a lived experience of the participants. There was already a similar study conducted looking at objective measures of sleep using actigraphy and similar metabolites. However, the literature is quite clear that there is a significant discordance between subjective and objective measures of sleep quality. And it's hard to say what is more important, but we feel that the lived experience of patients and what they tell us is what we want to ultimately end up treating for. So that was the rationale behind it. And because there's so many confounders to sleep, we excluded those with comorbid substance use disorders issues and mental illnesses. And this certainly increased our internal validity and increased the strength of our associations, although one can certainly argue that it's not truly representative of the population of women with HIV as a whole. Moreover, we know that different ARTs and ART regimens have different affinities to cross the blood-brain barrier, and we didn't account for specific ART regimens. And then we didn't account for other inflammatory conditions. As I mentioned early on, interferon gamma and TNF alpha are major determinants of IDO activity. So conditions, for instance, rheumatoid arthritis might contribute to the effect we're seeing. So in conclusion, we find that lower levels of 5-hydroxytryptophan and serotonin were associated with grey time dysfunction in all women, those with and without HIV. We saw that markers of increased IDO activation, both kynureninic acid and the kynureninic acid tryptophan ratio, were associated with daytime dysfunction in only the women with HIV. And that in future, we're ready to look at longitudinal associations to confirm these results and to see how or whether changes in these metabolites correspond to changes in daytime dysfunction. We're also interested in involving the gut microbiota into our analysis and seeing if this has an influence in daytime dysfunction and trying to mediate the role of inflammation in our sample. So in terms of acknowledgements, I'd first like to thank you all for listening to this talk, but most importantly, I'd like to thank the women in the WISE study, the Women's Interagency HIV study, for their contribution to the field. Then I'd like to thank my primary mentors at Hopkins, both Raha Daskeb and Leah Robyn, for facilitating all of this. And then to my mentors in Chicago, Kathleen and Audrey, and the various other important co-contributors to this work, as well as the Broad Institute for their Metabolomics and our various funders. And this work is still somewhat in its infancy. We're looking at this pathway across cognition and across motor control as well. So we're interested in it more broadly. And if anyone has any questions that they want to ask me at a later stage, my email address is there. Or if anyone has any suggestions, please feel free to email me and we can schedule a discussion on it. So thank you very much for listening, and I appreciate your time, and we'll open any questions.
Thank you, Eran. Again, excellent talk. Both of you guys have done a great job. Anybody have any questions?
So hi, this is Pim again. So Leah, were you yesterday also at the plenary where Grace Aldrovandi was presenting on particularly this pathway and some metabolic markers that were significantly affecting outcome in HIV infected or HIV exposed babies?
I was not. It sounds like I should have been.
Right. So so maybe you should. So they actually found two so one was called something 12-12-7. I have no idea what it was, not being. And the other ones were some other metabolic. But anyway, so I would basically say, you know, talk to Grace because they seem, at least at UCLA, also particularly interested in this pathway in HIV as well as HIV-infected kids. I think it was actually in part associated with breastfeeding mothers, but
no, that's, yeah, I think I got down, I forget when, yesterday in DC, but yeah, that's, Eran, we should follow up on that for sure.
Okay.
Oh, absolutely. Thank you, Pim. I appreciate that. That sounds interesting because tryptophan does pass through breast milk. And I guess the microbiome is also very important in terms of whether children are born via cesarean section or via natural deliveries. That might play a role in HIV too.
Anybody else? Yale, looks like you're gonna speak.
Hey, Eran, great job. And for Robyn's, both listen to your talk again after the ISNA. So as Eran, as we discussed, I said great job. And just I think if you can including the samples in the future study, including the people living with HIV is male, maybe more pervasive, right? For the K80 pathway, whether it's a true gender difference or not. And also, as we discussed, we use the echo-HV mice model. We also did the stool metabolomic. Very interesting correspondence is there. So I would like to be further discuss with you. Just one comment. Yeah.
Oh, absolutely. We need to have more than one discussion. We'll have a lot of discussions down the line. And in terms of male samples, we don't have male samples at the moment, but we're going to get a whole lot more male samples in the next. We just had a meeting this morning, and probably in the 9 to 12 months, we'll have more male samples, and then we can investigate sex differences there, and see if the TK pathway really relates to daytime dysfunction in men too. Although we know that sleep disturbances predominantly affect women, there still might be the pathway in both sexes.
And you'll also have the actigraphy as well. So not just the subjective, but the objective sleep data as well.
Yeah, absolutely. And the longitudinal analysis.
Yeah, so there's a lot of work that Eran will be doing. Any other questions? All right. With that, we will close our session for today and circle back next week. I think Cassie was having trouble joining. I think she fell off.
I'm back.
You're back. Of who's speaking next week?
So next week we have Suvendra Bhattacharya, and they will be presenting, sorry if I'm butchering any pronunciations in this title, miRNA export, transport, and uptake via extracellular vesicles in mammalian cells.
Awesome. So again, Eran, Robyn, thank you guys so much for amazing, I mean, honestly, just superb talks. And we will see everybody again next week. Bye, guys.
Thanks. Nice
Thanks,
job.
everyone.